Hello, my name is Erica Johnson, and this presentation covers the element copper and its role in environmental toxicology. After an introduction of what copper is, I will cover its common uses, to include copper's role in physiological processes, current environmental guidelines concerning amounts of copper in the environment, the pathways copper takes in terms of environmental impact, and a short case study. Copper is a naturally occurring element and essential mineral found in the environment. Like all minerals, it goes through a biogeochemical cycle as seen in this image. The biogeochemical cycle is defined as any of the natural pathways by which essential elements of living matter are circulated. This image serves as a model for the geological circulation and anthropogenic use of copper, from an article titled Earth's Anthropobiogeochemical Copper Cycle from Rausch and Gradle, 2007. Natural reserves of copper include the Earth's core, mantle, crust, biomass, freshwater sources such as glaciers, permafrost groundwater, lakes and rivers, ocean water, and the atmosphere, from Rausch and Gradle, 2007. This chart provides perspective into where the highest concentrations of copper can be found. Copper is both an element and an essential mineral. Copper typically exists in one of three oxidation states: Cu, Cu1+, and Cu2+, which is the most predominant. Its atomic number is 29 and atomic weight is 63.545. The chemical form of copper in the environment determines its bioavailability. An organism's uptake depends on the chemical form, environmental distribution, transportation method, and the organism itself. Because copper is an essential mineral and organisms have a natural ability to process and excrete copper, bioaccumulation is not an effective measurement of copper in the environment. Some factors that determine the bioavailability of copper include acidity and alkalinity, water hardness, cations and anions, complexing agents, and binding agents. Copper is rarely found in large quantities and is usually extracted from sulfide deposits rather than mines specifically built for the purpose of extracting copper. Common anthropogenic uses of copper include uses in infrastructure and housing, aircraft, generators and motors, treated lumbers, phones, and computers. Copper is also heavily used in the agriculture industry and is found in products such as insecticides, fungicides, and fertilizers. Though copper plays an incredibly important role in the modern society, it is vital to organisms as an essential mineral and critical in physiological functions. According to the National Institutes of Health for humans, these functions include energy production, iron metabolism, connective tissue synthesis, neurotransmitter synthesis, regulation of gene expression, brain development, and the immune system. Approximately 2/3 of copper in the human body is found in the skeleton and muscle. According to the Site and Life Organization, copper is the third most abundant dietary mineral. Enzymes that are dependent on copper include those whose purpose is to transport and transfer iron into hemoglobin, which carries oxygen through the bloodstream. These enzymes also develop collagen, which is important for both skin and bones and regulates histamine. There's also an example of primary natural sources of copper in the environment. Health risks of copper toxicity include liver damage and gastrointestinal symptoms, but is rare in healthy individuals with no hereditary defects. The most common source of copper toxicity is from consumption of water with high levels of copper. The average amount of copper stored in a human body is between 50 and 120 milligrams of copper, and the daily recommended intake for an adult female and male is 1,100 micrograms and 1,400 micrograms per day, respectively. Since copper is an essential mineral, organisms are able to excrete excess amounts of copper through natural processes. Currently, there are no biomarkers available that can accurately assess the amount of copper in a human. The EPA transitioned from a water hardness-based model to the biotic liquid model in determining the thresholds of copper in the environment. This method is tailored to a specific body of water rather than a generic model that excluded necessary parameters such as pH, temperature, alkalinity, dissolved organic compounds, and other pertinent variables. Under the BLM model, the EPA determined that 629 streams and rivers and 5 sediment areas sampled were not in compliance with standards in the United States. The water effect ratio method is a biological method to compare bioavailability and toxicity in receiving waters versus lab test waters. According to the EPA, the WR is calculated by dividing the acute LC50 of the metal determined in water collected from the receiving water of interest by the LC50 of the metal determined in standard laboratory water after adjusting both test waters to the same hardness. The hardness-based criteria concentrations are then multiplied by this ratio to establish site-specific criteria that reflect the effect of site water characteristics on toxicity. This chart exhibits the BLM input parameters required to determine whether or not a source of water is within acceptable limits for copper concentrations. This table outlines national primary drinking water standard regulations regarding amount of copper in the water supply. Copper is regulated by a treatment technique that requires systems to control the corrosiveness of their water. If there is more than 10% of tap water samples that exceed the action level, water systems must take additional steps. For copper, this action level is 1.3 milligrams per liter. Current environmental guidelines vary upon how the BLM is implemented. The BLM model can be targeted, meaning site-specific, or states can retain existing water hardness-based criteria. However, they are encouraged to transition to the newer BLM method, which is not actually enforced. As of 2022, the BLM model is still not enforced. State water quality monitoring programs vary on how and if the BLM method is implemented. As mentioned earlier, these programs can be either targeted, pertaining to a specific body of water, or states can retain existing water hardness-based criteria. Based off the examples previously provided, the most common pathways of environmental impact include anthropogenic processes, such as open-pit mines and refineries, copper manufacturing processes, agricultural practices, to include domestic use, and leather processing. Natural phenomena include volcanic processes, wind erosion, and forest fires. A case study titled Review of Copper and Copper Nanoparticle Toxicity in Fish suggested current knowledge surrounding copper and copper nanoparticles and their toxicity to fish is lacking in research. This article mentioned that there was a trend in significant amounts of copper near mines. Additionally noted is that heavy metals do not degrade with time, rather they absorb by sediments and aquatic life. Another difficulty in understanding toxicity parameters lies with the variances in the anthropogenic uses of copper. This study focused on the effects of fish organisms with wide parameters and focused on free cupric ions, which are used to deter fungal spores and are also the most toxic form of copper. The article also highlighted that most amount of copper is typically found in the gills, kidneys, liver, and skeletal muscles of aquatic organisms. Additionally, copper plays an important role in the red blood cells, nervous system, and immune system. As it stands, there is relatively little data available pertaining to aquatic species and the effects copper has in their respective environments. More research is required, but flora, fauna, and aquatic organisms are vital in being indicators of potential issues in the future. Since copper is both an essential mineral and element, it is difficult to determine the parameters and potential long-term consequences considering the variabilities in the environment, organisms, and the ecosystem. With all things considered, this makes it even further difficult to establish concrete results and with that standards to adhere to protecting and preserving the environment. This concludes my presentation, and thank you for your time.